So, on to chapter 13. Now, at first look, chapter 13 might seem very different from chapter 12, but I want you guys to realize that they're highly related, okay? It seems like we're going off on a different topic, but we're not. So don't do that. Don't separate that. And you'll see as I go through this lecture what I mean. Okay, so we're talking about heredity, genetics, same thing. So we just learned meiosis, which was making the sex cells, the gametes, and then those things will come together to make the offspring. And they're carrying the genes, the genetics here. And this, typically students enjoy this topic because it explains, why we look like our siblings or don't, or our parents or whatnot, and why we have this trait and they don't, or whatnot. It's pretty cool, okay? So there's also the constant argument, how much do genes affect who we are compared to the environment? And it's a mixture. You can look up different, papers, and it's still pretty debated. But lately, actually, the environment is getting a little bit more of support. I guess if I was going to say, I would say it probably in the past, I was believing that it was probably like 80% for sure genetics and 20% environment. Now, learning about epigenetics, and we'll talk about that a little bit in the future, but it's mostly a little higher level, but I do want you guys to be introduced to the concept. We're finding out that environment is affecting if our genes are expressed or not. So that I think I would have to say maybe it's now maybe more, like 70-30 in my opinion. But there's going to be scientists that have evidence. I'm just going by it, reading this here, reading that there, my gut feeling. You want to look at real research. Okay, but here's an example here with the Siamese cat. And in the colder environments, if the cat lives in here compared to living in Hawaii, the Siamese cat will have darker areas around their, what is it called? Extremities, thank you. And especially like their ears that are thin, right? And those blood vessels are right there. And doesn't this make sense? Why would it be better for the cat to have darker fur in a colder environment around these particular areas? Right, so we're going to get the sun's light, we're going to absorb that, black absorbs, and it's going to help them stay warmer, right? Whereas if they live in a warmer environment, they want to be lighter, right, for their environment. Okay, so it's definitely a combination. Just the exact percentage of how much does what, how much does the other is debated. Now, fun thing to study, is twins. And twins are, yes, and we need to study identical twins here. They tell us, they have the same genes, so any differences they have then should be from the environment. So they're interesting to study for this question. Now let's make sure we understand what type of twins. Paternal twins or fraternal twins, 2 eggs were ovulated, 2 separate sperm did the fertilization. Those are genetically as close together as normal siblings. They could have, their environment though is for sure similar, right? So if mom was, got sick during her pregnancy, that could potentially negatively affect both of them. So they do have a close environment, the paternal twins. However, identical twins, remember, 1 egg, 1 sperm. As soon as it got fertilized, it started doing mitosis, right? And it split into 2 separate little balls of cells. And they share the same sac, the amniotic fluid, the amniotic sac, and the same placenta, even though they each have their own umbilical cord coming from the bigger, same placenta. So those are your identical twins, the exact same DNA then. You guys with me on that? Now, just a teeny bit about epigenesis, what I was mentioning with the environment, is like in the last 15, 20 years, there's been a lot of information, a lot of research. They've been figuring out that, so here are identical twins. And because of environment, maybe what they eat, for example, certain genes could be being turned on, because remember, we're diploid. So maybe in this one twin, she's getting her dad's chromosome turned on. And the other one, maybe it's the mom's chromosome. So how the DNA is being transcribed, and it can be transcribed differently, can also then explain some of the differences in identical twins. Now, it's not the DNA that's making it different, it's that what gene is being turned on and off. We also know of randomness too, especially for females. So we have the two X's. You guys are evidence that we don't need two X's because you are working fine with one X. So what we did realize is that in females, in mammals, one of the X's is randomly silented. It's actually kind of balled up. It's called a bar body. And so it can't be transcribed. The DNA is not going to open up for transcription. So which one was silented? It's random in each cell. So there can be difference that way too. So anyway, that's kind of an environmental influence. With the other one, it's called, one thing of epigenetics is called methylation. So we're finding that people that have a better diet, that have eat more variety of foods, that definitely have more methyl, which is CH3, basically, in their diet. They're able to shut down some genes. They're basically able to like clog or stop genes from being expressed that could have negative responses in the body. So anyway, they're learning more and more about that, which is pretty interesting. Okay, so moving on to Mr. Gregor Mendel. So this is going to be your scientist of the unit here. He lived in the 1800s. He is most famous for his genetic work, but how he did his genetic experiments. He used huge numbers in his experiments, so lots of trials, which is always good. He also got lucky in one of his choices, and he chose peas, and we'll hear more about that in a second. He was a monk. He had some background issues like Darwin, where he wasn't sure what he wanted to be. If I remember correctly, I'm pretty sure his dad was a doctor, and they wanted him to be a doctor, and this is the same case with Darwin too. And he tried, I think, and then it didn't work out, so then he decided to be go to the church. Now, this allowed people to have an occupation there that they were being provided for and doing their churchly duties, but it also gave them free time to read and write, right? So a lot of interesting people have come out of, the monasteries and in the church, great writers, and commonly also some great scientists, okay? So he was in charge of the garden, and remember, that's not a hobby in the 1800s, right? He's providing food. The better a gardener he is, the more food they're going to get, and probably for trade as well. So let's listen to this.
In the 1860s, Gregor Mendel used garden peas to study heredity. Peas are very easy to grow, and Mendel could study 9 generations during his 8 years of experiments. Peas are also self-fertilizing. The petals of the pea flower completely enclose the reproductive organs. As a result, the pollen from the anthers falls on the stigma of the same flower. Pollen tubes grow down through the female reproductive organ to the ovules in the ovary. The ovules develop into seeds, and the ovary wall develops into the pea pod.
So you maybe have heard the debate, is that a fruit or a vegetable? In science, fruit is coming from fruiting bodies, which means that's where the offspring are coming. So anything with seeds is really defined as a fruit. Now, we kind of define it differently in the grocery stores, right, or at home. Tomatoes are fruit. A lot of people would say maybe they're vegetables, right? Vegetables, that vegetation part of that word, means stems and leaves and roots, okay, not the fruiting body, the flower. Also, I know I went over the pea flower anatomy with you guys, but the model doesn't show the big petals. It'd just be kind of, I guess, too big for this model. But when he chose the peas, because they self-fertilize, they're all clones, so he knew what he had. A purple flower always had purple flower babies. So he ended up working with, we call them true bred or homozygous, purebred. So he ended up getting to work with things that didn't have a lot of variety in them, and then he was able to do successful crosses. Okay, so also I want you guys to realize the flower, once fertilization has occurred, it's going to turn into the fruit of whatever plant it is. Now, you maybe never put this together, but you could see here that pea. So down here where the eggs are, as soon as fertilization's occurred, pollination in this case, right, for the plant, the petals will start shriveling up. They've done their job and fall off, and that ovary, as the eggs develop in there, will grow, and you can see it on the picture here, into the fruit. So peas, right, are seeds. Pea pods, the whole fruit. Okay, so I mentioned the true breeding, and he did crossbreeding. I'll explain that in a little bit. And he figured out that he thought there has to be something that's being passed on from generation to generation. He didn't know how, but he called them factors. He thought the parents were passing on factors, pieces of information to their offspring. That's true. We now know them as what? What did we name them? Genes, okay? And then your definition of gene, segment of DNA, whose sequence of nucleotides codes for a specific functional product. What are we talking about? What are the functional products? Oh, you're scaring me. DNA codes for RNA. RNA codes for proteins, which are made of amino acids, right? So the differences are simply because of the different proteins that are being made by our individual DNA. Same case with the peas. Okay, now, there is a lot of vocabulary. You saw on your learning plan, I gave you a vocab list for both chapters. On the learning plan, I did notice there was a slight typo. Well, there's a couple typos. I really just wanted to get that to you guys so you had the vocab list. But on the back side for the, let me see it, the essential questions on the back, say chapter 12, that's supposed to be chapter 13. Chapter 12's essential questions are on this side. So if you looked at your learning plan, I tried to set it up 12 on one side, 13 on the other. However, on the back side, learning targets all together for both chapters. Okay, alleles. Alleles, just the possibility that the gene will code for the different protein. So with the pea plants, purple petals versus white petals. Green peas versus yellow peas. For us, hair color, skin color, all the different variety, nose shape, handedness, okay? Okay, you know how we like to then reflect on, well, what do bacteria do? What do the prokaryotes do with this? Just a little slide here. Remember, they have one chromosome, circular, and 90% of that DNA is translated. The genes are expressed. That's not the case with ours. We have a lot of DNA that we don't even know what it does. Okay? But they also have those plasmids. Remember, we're talking about chapter 13, but think back on 12. Conjugation. The bacteria can kind of do a little sexual reproduction, just partial, by passing on their plasmids. So plasmids are just little teeny circles of DNA that have a couple genes on them. We'll work with some plasmids that humans engineered in the future when we do the transformation in a few more weeks. Okay? Okay, let's talk about chromosomes. So the chromosomes, you know, when we first, you know, we could see chromosomes. Didn't you guys do a lab, the mitosis lab? And I tried you to ask to count them. It was pretty difficult. Remember this? but you could see them. Now that's with our microscopes. Okay, a lab can have a light microscope that goes up to 1,000 times, so double kind of what you saw, and you'd be able to see them a lot better. Also remember the chromosomes to be folded under themselves. That's a 3D world, and you are looking at it in the 2D way. Also, those cells were killed, so they were like kind of frozen whatever they were doing at that time. But anyway, so this picture here with the fluorescent dyes, it's just a random bunch of chromosomes. They are X to me, so I bet this is in prophase 1, right? And anyway, so the hard part was studying these things and then figuring out, hey, they're in pairs, okay? And we could use all different dyeing techniques to figure that out, staining patterns, and we saw they're true homologs. They dye the same, they're the same shape, they have the centromere in the same spot. These are your homologous chromosomes. Now, when we take the picture of them and do, copy drags or cuts and drags, we can reassemble them in an organized manner where we have them matched up. They're never going to match up like that. They're not all going to get in a line, right? But we could take a picture and then we can cut that image and move them around. In the old day, this was done with film, with scissors. Now we're doing it all on computers. We call that study of an organism's chromosomes a karyotype. And there's a picture of a karyotype. So when people get amniocentesis, they're worried maybe their baby had something wrong with it. A mom and dad could get amniocentesis, and they'll be looking at the baby's chromosomes to see if there's any huge chromosomal mutations. Is this a boy or a girl? It's a girl because she has 2 X's. And you might say, Coop, they don't look exactly the same to me. One of these X's is longer than the other. You have to remember they were in cytoplasm. They're in fluid. So they can get smooshed down, right? They could get pulled and stretched out depending on when the picture was taken. You see that they bend and all that. But the banding, the staining is amazing how you can really easily pair them up. We numbered them from the largest pair to the smallest pair, and then the 23rd pair, if you want to call it a pair, is your sex chromosomes. Makes sense for the female, and then the male, they wouldn't match. Okay, then we'll move on here to the next class to this.